apologize that the video isn't working, but just please take a minute and take a deep breath. Listen to the sounds. Hi again. So good. I am sitting again because today we are discussing. Oh, this isn't working. Okay, today we are discussing Hardy-Weinberg equilibrium. And Hardy-Weinberg equilibrium, there's a lot of stuff happening. There's a lot of calculations going on, a lot of steps. And I want to make sure that I'm not skipping anything because I don't want it to be a confusing thing for you. So I am sitting because I don't have the best vision in the world. And if I am not in front of my computer, I can't see my notes. So back to sitting now, if you even noticed. And I don't know why this isn't working. Now we're rolling. Okay. Getting into discussion of segregation and Hardy-Weinberg equilibrium. And Hardy-Weinberg equilibrium might be new for a lot of you. We're going to discuss kind of what it actually is and what it means, and then do some practice problems. Some important things to start with is the concepts of allele frequency and genotypic frequency. Allele frequency is how often, that's the frequency, an allele occurs in a gene pool. How often an allele occurs in a gene pool. And its definition kind of has its name in it. So how often, that's frequency. And the gene pool is the population. When we're discussing all of these things about evolution, we're talking about an interbreeding population. So that would be a gene pool. So allele frequency is how often an allele occurs in a gene pool. And the maximum frequency for the entire everything, the maximum frequency of any allele, the maximum frequency that we're ever going to get to is 100%. So everything we do is out of 1 or 100%. And you'll see alleles, the allelic frequency, you'll see this expressed as a decimal. So like you'll see a frequency of 0.2, for example, or you'll also see that expressed as 20%. And this is something you're really familiar with. It's a percentage or it's a decimal. Kind of the same thing. If we look at my imaginary mouse population, you can see that there are 3 phenotypes. There's the white mouse, the purple mouse, or the green mouse. If you're not able to differentiate those colors, the first group is white, the middle group is purple, and the last group is green, and that's not particularly significant. That's just indicating that they each have different phenotypes. If we're interested in figuring out what the allele frequency is for this population, we need to remember that every genotype that we're looking at is composed of 2 alleles. So all genotypes are composed of 2 alleles. Again, this is something that you guys know. In our imaginary population here, our population has 8 mice, so 8 individuals. Okay. So if we have 8 mice and 2 alleles, that means that there are 16 copies of this gene in this population. because each allele represents a copy of the gene. They might be slightly different copies, but it's still the same gene. So in our population, we have 16 total copies of this gene in this population. If we want to figure out the allele frequency, this is how many of each allele there are out of this 16, right? There's 16 copies. How many of them are each thing? So all we have to do is count them up. All of our alleles that are homozygous A1, all of our, genotypes that are homozygous A1, there are going to be 2 A1s. So there's 4 mice that are homozygous A1, so that would be 2 times 4. We've got 8 A1s there. Here we have a heterozygote, so there are 2 A1s here. There are 0 A1s here, so 8, 9, 10. So we've got for A1, 10 copies. And then for A2, this individual has 2 copies of A2, or these 2 individuals between them have 2 copies. Each of the A2 homozygotes, there are 2 individuals, and again, times 2. So we get 4. So 4, 5, 6. So we have, A2, we've got 6. And to get our percentages, we take that out of 16. Okay, and then you can just do the math to get the percentage, to get the decimal, and that is your allele frequency. Pretty straightforward. You're just figuring out how many of the alleles are there out of the total. This is important because, as you know from genetics, when we're making babies in a diploid species, in a sexually reproducing species, we go through a really important process called segregation and independent assortment. And what this means is that every single offspring can have a different genome than its parents. Remember that which direction each of these copies, each of the sister chromatids gets pulled is random. So if we have a heterozygous individual, then this can all get shuffled, right? So we have that in our first round of meiosis, and then again in our next round of meiosis, we split again. That's random, which direction each of these goes. And so just from this cell on the left, we get 4 different possible arrangements, right? We can get a lot of variation in the arrangement of the alleles in the resulting population. Just that's a reminder from genetics. And so what all of this sort of that I've been talking about, that I'm introducing you to the concept of Hardy-Weinberg equilibrium. I have that definition here somewhere. Oh, that's next. Okay. So let's imagine that we have a population and the parental generation is all homozygotes. We have 100, where 50% of the population is an A1 homozygote and 50% of the population is an A2 homozygote, and this population interbreeds. And we want to think about what's the probability of their offspring. What's the probability of each of these potential combinations? So we can of course get offspring that are A1 homozygotes, just like one of the parentals. We can get offspring that are A2 homozygotes, just like the parentals. But then of course we can also get heterozygotes. Each of these possibilities, there's a half probability for each. The probability of one of the offspring getting an A1 is 1/2, because half of the population only carries A1s. Then we've got a 1/2 possibility because the A1s can mate with each other, right? And they would donate an A1, half of the population. So each of these, because half of the population is each thing, you have half probability of receiving each of these alleles. The offspring have half of the probability. We multiply those, as we learned, in probability. So there's a 1/4 probability of each of these combinations of alleles in the offspring. If we consider the fact that the heterozygotes, doesn't matter which direction we write those are the same genotypes, what we end up with is a quarter in the F1, in the F1 generation, a quarter are going to be homozygous A1, a half are going to be heterozygotes, and a quarter will be homozygous A2. And this is just review of concepts that were covered in genetics. If you're not remembering this, maybe do an A1, A2 Punnett square to remind you how that happens. Okay, so it looks like we've got a change, right, from our parental generation to our F1 generation. Our parental generation was half A1 and half A2. Our F1 generation is only a quarter A1 and a quarter A2. So what's changed then is our genotypic frequencies. From parental generation to F1, we get a change in our genotypic frequencies. What has not changed is our allelic frequencies. So check this out. Half of that half is a quarter, right? So a quarter of the alleles in those heterozygotes is A1. A quarter of the alleles in the heterozygotes are A2. So now if we take our allelic frequency, it's exactly the same as the parental generation. One half of our alleles are A1. One half of our alleles are A2. So we did not get a change from the parental generation to the F1 generation in our allele frequency. Genotypic frequency changed, allelic frequency didn't change. This is what we're talking about when we're talking about Hardy-Weinberg equilibrium, is the allelic frequency didn't change. Hardy-Weinberg equilibrium then. I always forget how to spell Weinberg. Weinberg. Hardy-Weinberg equilibrium. So what does Hardy-Weinberg equilibrium all about? Relative proportions so the proportions relative to each other of genotypes in a population will not change, when segregation is the only factor that changes genotype frequencies. Frequencies. There's an I in there. Okay, so come over here. Okay, so you might be thinking, but wait, we were just talking about alleles, which we were. So why does this say genotypes? The reason that it says genotypes is because when a population has reached Hardy-Weinberg equilibrium, that's when the genotypes, the relative proportions of the genotypes aren't going to change. So in my earlier example here, which one is that showing you? Okay. From the P1 to the F1 generation, we saw a change in the genotypic frequency, but we did not see a change in the allelic frequency. That stayed the same. Now, in this F1 generation that I used as this example, this population has reached what we call Hardy-Weinberg equilibrium, because moving forward, in the absence of anything but segregation, if segregation is the only thing influencing this population, these genotypic frequencies are not going to change. They're not going to change again unless some other force besides segregation acts on them. Okay, so you might be thinking, well, that sounds kind of unrealistic. I always hated Hardy-Weinberg equilibrium when we would study it because we would be taught, oh, you have to learn how to calculate Hardy-Weinberg equilibrium. You have to learn what it is. But it only applies when nothing is happening. It only applies when there's no natural selection, when there's no drift, when these things that we've been talking about, when they're not happening. And I always thought, why do we have to learn this? Well, the reason that we learn Hardy-Weinberg equilibrium is because it's what we call, it'll come back, come on, goodness gracious, guys. It's what we call a null model. So Hardy-Weinberg equilibrium is the model against which, it's the expectation against which we compare our real life data. And you can absolutely have places in the genome that are not experiencing drift, that are not experiencing natural selection. And they will be in Hardy-Weinberg equilibrium. And so we can compare our real data to the expected Hardy-Weinberg equilibrium, which is our null model. So I wanted to explain that to you because it was always a major source of frustration for me. So let's talk about this whole Hardy-Weinberg equilibrium thing. We're going to get to alleles. We're going to start with phenotypes. We're going to get to genotypes. We're going to start with phenotypes because it's easy, I think, to start there, to first just think of this in terms of a phenotype. So if we have our imaginary cute little blob population here, and P, or the purple blobs are representing the dominant phenotype, whereas the yellow blobs are representing the recessive phenotype. And in Hardy-Weinberg calculations, the P represents the dominant phenotype and the Q represents the recessive phenotype. What we're going to need to do, as I sort of showed you earlier, is we're going to have to count these guys up. So let's organize them, make that a little bit easier so that we can figure out what are our ratios. We want to figure out what's our frequency. What's our frequency of P? What's our frequency of Q? That's our question. And sort of a central thing to always keep in your head. P plus Q equals 1. Remember, the probability of an allele having either the dominant or a location, a locus allele being either the dominant or the recessive equals 1. We're sticking here with a two allelic system. You guys know from genetics that there are many systems where there are many more than two alleles, but we're sticking with a two allele system for the purposes of understanding the concepts of Hardy-Weinberg equilibrium and learning to work Hardy-Weinberg problems. So this is a two allelic system. If there only exist two alleles, then P plus Q, the probability that a one locus, one allele, is either going to be P or Q is 1. Right? 100%, it has to be one of the two. So for a phenotype, the way we figure this out is we take our total population. In this case, our total population is 20. We count up our P's, we count up our Q's, we divide those by the total population, done and done. We get what percentage of our population, what proportion of our population is purple, what proportion of our population is yellow. We got 7.7, 0.3, boom, they equal 1. Okay, hopefully that is super clear. We're just taking a percentage, right? When we get into genotypes, however, our genotypes are all composed of two alleles, and we can have heterozygotes. So this is where we start, we need to bring in the concepts of the Hardy-Weinberg equilibrium, and we're going to pull in that P and Q terminology. Excuse me. If you see here, our homozygote has two P's, our heterozygote has a P and a Q, and our other homozygote has two Q's. So in this case, our purple homozygote is our dominant, and our yellow homozygote is our recessive. So allele 1, P plus Q has to equal 1 at that one allele. Allele 2, P plus Q has to equal 1 at that one allele. This is something that confused me for a while, and if that absolutely totally makes sense to you, awesome, but I'm going to step through it to prove it to you. So that's not good. Hang on, guys. I need to fix that. There's way too much happening on that screen. It was supposed to be animated. Wait a second. Okay, that's better. That's a little bit less overwhelming, I hope. Okay, so we're going to take an imaginary population. This is our imaginary population of 20 individuals. And if you want to pause, take a minute to count that up, please feel free to do so. Our imaginary population of 20 individuals. And so we're going to sort of figure out, let's step through this to prove to you that allele 1, p plus q equals 1, and allele 2, p plus q equals 1. So using, again, our same dominant recessive situation as I already showed you. We already discussed, I guess, would be the way to say that. Our yellows, we know, are recessive. If our yellows are recessive, then in order for them to be yellow, they have to be homozygous recessive. So we know that our yellows, all of these yellow individuals, are homozygous QQ. 17 of them are homozygous QQ. For the purposes of this demonstration, let's imagine that 2 of our dominant individuals are homozygous PP, or our purple individuals, I apologize. 2 of our purple individuals are homozygous PP, and 1 of them is a heterozygote. So it has a P and it has a Q. Just checking to make sure you're able to see everything. I shrunk myself, made myself a little smaller. Okay, what's going on at allele 1? So at allele 1, that would be our first position. P plus Q, we're trying to demonstrate that that equals 1. So at allele 1, the first position, 3 individuals, the first position is P. So let's see, I'm going to show you that. So for these 3 individuals right here, the first position, so allele 1 is P, because there are 2 that are homozygous P and 1 that's a heterozygote. First position is P. Okay, so then at allele 1, at that first allele, how many are Q? Well, every single one of those homozygous recessive individuals, their first allele is going to have to be Q. So 17 of them are Q. There's 20 individuals in the total population. We divide by 20. Boom and boom. We get 0.15, 0.85. Add those up, you get 1. Let's do allele 2. Allele 2, again, P plus Q equals 1. At allele 2, at our second position, there are only 2 individuals that have a P there. Whereas there are 18 individuals that have a Q in that second position. We divide by 20 once again, we get 0.1, 0.9, and that equals 1. Why is that also significant? Well, because this P plus Q equals 1 at allele 1, P plus Q equals 1 at allele 2 is exactly how we get the Hardy-Weinberg equation. This is how we get the equation. That's how you're going to solve these problems. Allele 1 and allele 2. In order to get a genotype, you need them both, right? You need allele 1 and allele 2. When you get an and, you multiply. So we're multiplying probabilities here. And what we're doing is we're just using the distributive property or the FOIL method. Hopefully you guys remember the FOIL method to get this equation. So remember, the FOIL method is first. So for allele 1, we've got P is first. So we go first to first, and I'm going to use purple. So here, first. I hope all that stuff we did in genetics, you're like, oh, that's awesome and useful. So P times P is P squared, first. Then we do inside. So Q is inside P. So we get P and Q. Sorry. Inside. Then we go outside. P to Q. Again, we got another P and a Q here. And then last. Do I have any more colors left? Last. So this is just like our FOIL method that we used in genetics. So now we have 2 Qs, that's our Q squared. Just like with our heterozygotes, P times Q is the same as Q times P. So those together become 2PQ right here. P squared plus 2PQ plus Q squared is the Hardy-Weinberg equation, and this is what we're going to be using from from here on out. And that's how we get that equation. So this whole p squared plus 2pq plus q squared, we get it from the two alleles, from multiplying what's happening at those two alleles, because you have to have both alleles to get the genotype. Okay, so what's happening at each allele for the entire population is how we get this Hardy-Weinberg equation. This is why it has some things are squared and some things are multiplied. Okay, all right, so this is our genotypic frequency equation. Once a population has reached Hardy-Weinberg equilibrium, it should stay there unless we have other disturbing factors, unless we have selection, unless we have drift happening, right? Mutation is another thing. And we're going to talk more about those later. All right, genotype frequency, Hardy-Weinberg equation is p squared plus 2pq plus q squared. P squared is the homozygous genotype, the first homozygote, usually the dominant, because we're usually talking about a dominant recessive system, but you guys know from genetics, sometimes we don't have a simple dominant recessive system. The heterozygous genotype, and then the other homozygote frequency. So let's work through a problem. How long is this video? This is 26 minutes long. We're going to stop there before we do a problem. Take a breath. Pomodoro technique. I'll see you back here in a minute. Or not a minute. That's okay too.